do. I know which one you're talking about. Right after fifth.
All right. Please keep in mind we took that prerequisite exam on Monday. Okay. Where we see the biggest crossover in content is with these earlier chapters. What we are going to cover today, as well as next Wednesday, and then the following week in talking about microbial growth. There's a lot of crossover content. There's a lot of content you should have had in some other biology class. If a lot of this stuff looks foreign to you, I highly suggest you read those chapters in the book closely, because you need to understand the differences between prokaryotic cell structure and eukaryotic cell structure. Almost every chapter that we cover from this point forward is going to come back to those differences in cell structure types. When we talk about ribosomes and what they do and cellular components and what they do, you need to have those down cold. Otherwise, when we get later into the course and we start talking about antibiotics and how they work and which ones are acting on ribosomes and which ones are not, it will then be complicated for you to understand, okay, if it's acting on a ribosome, that means we're affecting protein production and so on and so forth. Okay, so yes, a lot of this content should be content you've seen before. We want to make sure that we're refreshing our memory on all of this for what's going to happen later on in the course. We're going to start by a little bit of a discussion on scientific inquiry and the scientific method. Keep in mind what we talked about on Monday is that we have this thing called spontaneous generation. And spontaneous generation was basically this idea that living matter magically came about from non-living things. Okay, and we talked about the fact that because we couldn't see these microorganisms, they're too small to be seen with the living eye, that nobody believed they were there. Everybody was relying on religious beliefs and superstition to understand that this is what's going on. It's not just some sort of tiny little living thing that I can't see. Okay? Now we understand we use the scientific method to explain these phenomena and how they work. So this particular process, the scientific method, goes through a specific pattern of making a hypothesis. So we observe something, we observe a natural phenomenon. Our hypothesis is what we think is happening. We then make predictions. If I test this, this is what's going to happen. So we're thinking about our experiment and trying to understand, if I do this, this is what I expect will be the outcome. You will actually perform the test. You will take those results, and then you need to make conclusions from the results. We are doing this process in each and every lab, okay? We are looking at it and understanding, okay, what do I think will happen? There was a pre-lab question that said, what do you predict those results are going to be on that plate? What do you predict is going to be that condition that's going to result in the most reduction in types of colonies that are produced? We want you to think about what you think is going to happen. We've now performed the test. We're going to let it grow. When we come back next week, we'll be looking at results. And then you'll be using those results to make your conclusions. And the conclusions you will be tested on in the post-lab part of the lab. Okay? So you want to understand, yes, it seems like a very basic process. You learned about scientific method when you were in the second grade. But we're still going through that process. We want you to think about it. These lab questions, the pre-lab and the post-lab, are really there to make you think, okay, what's going on and how does that work? When it comes to the lab component, you want to make sure you're reading through the labs before you open that pre-lab, before you do the pre-lab. I heard someone before class go, well, I half-assed that pre-lab, so this will be fun, okay? Number one, I'm standing right here, okay? Number two, please understand that this is going to, that's going to be reflected in your grade, okay? You want to make sure you're understanding the components. You want to make sure that you are able to apply the components, okay? Because if you're not able to apply the concepts, when you see it on an exam question, it's going to look foreign to you. You're not going to be able to complete that thoroughly, okay? All right, so I guess I just stated what was on the slide, okay? If we're going to apply the scientific method to what we're doing in lab, we've got to understand the components. We've got to understand the parts we're looking through. If you read that lab manual and you're like, okay, there's a word here I don't know, Google it, okay? That's where you get out your phone and you plug it into dictionary.com or something like that and figure it out. You don't want to go, okay, I didn't understand what I just read, so I have no idea what I'm going to do in the lab. Try and apply your knowledge or try and figure it out. Last class, We talked about prokaryotes versus eukaryotes. And I keep saying this is going to be a common theme throughout the course, okay? So if we had to write out differences between prokaryotes and eukaryotes, what differences would you say there are?
Single-celled for prokaryotes.
Single-celled for prokaryotes. Okay, no nucleus. Wait, what'd you say? A cell wall.
There's no membrane-bound organelles.
No membrane-bound organelles. They're all living. Which ones are smaller? Prokaryotes.
They're more simple, like that.
Okay.
Whereas the eukaryotes are more complex.
Okay. How do they divide? So these guys divide by a process known as binary fission. What does their chromosome look like? It's typically a circular chromosome. And there's usually just one of them. What about the eukaryote side? What's the opposite of a circular chromosome?
Linear, like that.
Right, linear chromosomes. And there is typically greater than one chromosome. What process do they use to divide?
Mitosis.
Mitosis. Mitosis for all those somatic cells. Meiosis for the germ cells. We had single-celled under prokaryotes. What will we write under eukaryotes?
Multicellular.
Or single-celled. So these guys can be single. So there are some that are going to be single-celled. Or they can be multi-celled. Do they all have a nucleus?
Yes.
Yes. Awesome. What about a cell wall? Here's some no's. Some do and some don't. What eukaryotes would have a cell wall that you would think of? Plants. So plants have a cell wall. Most of the other eukaryotic organisms do not. So some have a cell wall. We said no membrane-bound organelles.
These do have some membrane.
These ones do all have membrane-bound organelles. Okay, so ER, Golgi, all of those are included in this one. Those were smaller, these are larger. And if the prokaryotes had a simplified cell structure, these guys have a more complex cell structure. And we just detailed that with membrane-bound organelles, nucleus, those components, okay? There are lots of differences between prokaryotes and eukaryotes. If you learned about histone proteins in biology, DNA is wrapped around these little histone proteins, kind of like beads on a string. The histone proteins help to keep all of the DNA organized inside the cell. Prokaryotes do not have histone proteins because they have a single circular chromosome. It's typically a much smaller piece of DNA because they have fewer genes, and therefore they don't need those histone proteins to keep it tightly bound. Okay? I heard membranes, okay, cell membranes. We're going to talk about those. Both prokaryotes and eukaryotes are both going to have a cell membrane, okay? And how they function is actually really similar between the two. There are lots of similarities here. There are lots of differences. We're going to really talk about exploiting those differences throughout the course of the semester. Everybody got this down? Okay. All right, so what does a prokaryotic cell look like? This particular figure gives you a lot of different components that a prokaryotic cell could have. It's not saying that every prokaryotic cell looks like this, because they don't. Not every prokaryotic cell has to have all of these components. So this is a picture of one type of prokaryotic cell to help you understand the different types of cellular structure. How is this guy oriented? Now we said more simple cell structure, okay? There's not a lot going on inside. Inside that cell, you've got a cytoplasm, that kind of gelatinous matrix that suspends everything inside the cell. You are going to have what's known as a nucleoid region. That nucleoid region is where the DNA is found. Keep in mind these guys do not have a nucleus, right? So there's nothing bound to hold on to that DNA and to keep it into one spot. So instead what they have is called a nucleoid region. It's a region in the cytoplasm where the DNA is found. You then have the chromosome structure itself. The other thing that's inside this cytoplasm are all of these little pink dots. All of those are ribosomes. What did we say the ribosomes are used for? Protein. Those are going to be the sites of translation, which are going to be utilized for protein production inside the cell. These guys have no other membrane-bound organelles. Ribosomes are not a membrane-bound structure. So when we said a difference between prokaryotes and eukaryotes is no membrane-bound organelles versus membrane-bound organelles, that does not include the ribosomes because ribosomes are not bound by membranes. Okay? So that's why they're kind of exempt from that particular list. All of that is in the center, and that's it. We have cytoplasm, we have the chromosome, and we've got those ribosomes. That's it. That's all that's inside these cells. Then you start to move to the external layers. The external layers are really important for a prokaryotic cell. You are going to have the cytoplasmic membrane first. That's going to be the innermost layer. The cytoplasmic membrane, you will also see abbreviated cell membrane. They are the same exact thing. The cell membrane is really good at making sure that nutrients are going in, waste is going out. It's just kind of a slight barrier to kind of keep the cytoplasm contained. You then have the cell wall. On a prokaryotic cell, they're going to have some form of cell wall. Almost all prokaryotic cells have a cell wall with a few exceptions mixed in. Bacterial cells specifically have a compound in their cell wall known as peptidoglycan. The cell wall made of this peptidoglycan is what provides a really rigid structure to these prokaryotic cells. This is why this particular cell looks like a nice bacillus shape or a rod shape. Other prokaryotic cells can take on a circular shape, which is known as a coccus shape. The cell wall determines the shape of those organisms. Your cells don't have a cell wall, okay? So their shape is really plastic. It's going to change over time, and it's going to depend on what's next door. Epithelial cells, as they're all grouped next to one another, they're pinching each other together, they can take on more of a square form. Your blood cells, on the other hand, are really nice and round. They're floating, okay? So our cells don't have a firm cell structure to them or a defined shape all the time. These guys do because of that cell wall. You can get additional compounds that are outside of the cell wall that have various functions for an organism. In this case, this particular figure is showing you a capsule. A capsule is more of a gelatinous coating outside a cell that is going to help with attachment. It's also going to help with virulence. And virulence means how easy it is for an organism to get inside your body and stay inside your body, kind of evading the immune system, so how long it can stay active. So capsules and other things that are on the outside typically are going to function with some part of the organism's growth, some part of their life cycle. Lastly, we have these extra appendages on the outside, and what you will notice is we've got a term down here called pili, and we have one up here called pilus. Those are referring to the same exact structure. Pili is plural, pilus is singular. These basically look like long little fibers that extend from an organism. These long little fibers can help with movement. They can also help with attachment for the cell. And lastly, what you see on here is a flagellum. This is very similar to a eukaryotic flagellum. This is going to move and rotate to allow those organisms to move forward in an aquatic environment. Remember, not all organisms are going to have all of these structures. So some of what you see on this figure are absolutely required, and some are going to be more optional. A flagellum, for instance, is an optional structure. Bacteria or other prokaryotes that live in aquatic or watery environments need to be able to move around that environment so that they get food access or access to sunlight if what they do is photosynthesis. So a flagellum is really necessary. An organism that lives in the soil, on the other hand, doesn't really need a flagellum. It's not going to be able to do anything with it. That structure is basically going to be useless. So we're going to understand or begin to go through which structures are required, which structures are not. This is a table. I like this table from the textbook because this table helps you to understand which structures are internal, which structures are at the boundary, and which ones are extracellular. And then, of course, you've got the characteristic or the function that's on the right. You want to be familiar with the cell structures themselves and what they do, okay? So a few of these internal structures. All I'm going to do at this point is break them down a little bit more. We've kind of already roughly went through them. A lot of these you have heard of. A lot of these you know what they do, so that's nothing new. Remember, we have required structures. We have non-required structures. Something like a chromosome is essential for life. Ribosomes are essential for life. The chromosome, remember, is going to contain the DNA or the genes for the organism. Without that genetic code, that cell is dead. Every living cell needs to have some amount of DNA or RNA present depending on what it is. So chromosomes are absolutely required. Ribosomes are required as well because ribosomes, remember, are going to be responsible for making protein. If you remember back, you have this central dogma of biology. That central dogma says DNA gets taken into RNA, which then becomes protein. Inside every living cell, they've got to be able to complete this process. What is the process called that takes DNA to RNA?
Transcription.
Transcription. And what's the process called that takes RNA to protein? Translation. You want to roughly understand how those processes work, okay? That is one of those prerequisite concepts. On Blackboard, there is a tab that's called Course Resources. There's a folder in there that's all got resources on transcription and translation. If you're like, I don't remember any of those, at least watch a short video on how those processes work. You can go to YouTube, you can go to Khan Academy, some other site, so that you're just, I understand the process of taking DNA to RNA and RNA to protein, okay? Because we need to do this, that chromosome and the ribosomes are going to be required structures for every living cell. The rest of the structures that are on this list are totally optional. Cells that have them typically have some selective advantage. They typically get ahead in some way that may allow them to survive longer depending on the environment. So organisms that like a watery environment may have the ability to survive when conditions dry out a little bit. Organisms that like a warm environment may be able to go into a dormant state when it gets really cold out. There's a lot of cell structures that these cells can have to help them survive. Don't just think survive inside the body. It could be surviving inside the environment as well. So for example, a plasmid is on the list. Plasmids are independently replicating pieces of DNA that give these cells additional genes, okay? You can have storage granules. Those are basically little storage components inside the cell. It basically looks like a walled off area where they can store nutrients for later, basically, okay? And then we'll talk about endospores as well. Endospores are a dormant cell type. Cells that experience harsh environments where they would normally die. If they're capable of forming an endospore, they go into that, through that sporulation process, they form the endospore, and that allows them to basically sit completely dormant until they're capable of growing again, okay? And then they go back to being a living and dividing cell. We making sense? Some of this coming back to you? Yeah, okay. Remember that chromosome. So for these bacterial cells, that chromosome is going to be sitting in the nucleoid space. It is typically a single chromosome in a prokaryotic cell. Notice, remember, we're talking about science here. We're talking about living organisms. Not every living organism is going to conform to these particular details here, but for the most part, all of the bacteria that we're going to talk about in almost all prokaryotic cells are going to have a single chromosome. Typically, this is made of some sort of double-stranded molecule. There's a reason here that this says molecule and doesn't just say DNA, okay? You've been taught that DNA is double-stranded, RNA is typically single-stranded, but that's not always the case. What makes DNA is the bases that it's made up of. Those A, T, C's, and G's, okay? That's what makes it DNA, are those four bases. The A's always pair with the T's, and the C's always pair with the G's. What makes a molecule RNA is that instead of having the T base, it has the U base. So now we still, A pairs to U now, and G still pairs with C. That's what makes it RNA. Not the fact that it's single-stranded or double-stranded. So yes, this says a circular double-stranded molecule because there are a fair number of prokaryotic organisms that have this circular double-stranded, but it's double-stranded RNA for its genome and not double-stranded DNA. That RNA, having that circular RNA, just makes it easier for that cell to make protein from in the end. Some of those are going to have a reverse transcriptase that will allow it to make DNA if it wants to. There's a lot of variety that happens, but typically the chromosome is going to be a double-stranded molecule. Most of the chromosomes are going to be double-stranded DNA, especially every organism that's capable of infecting your body. Most of those are going to be all DNA-based organisms. We're not going to see any that are going to cause human disease that are going to have an RNA structure. It's going to typically contain all of the genetic information for that organism, okay? Your plasmid, on the other hand, remember, is kind of an independent piece of DNA. It's typically roughly 10% or less the size of the chromosome. It replicates completely on its own, and it typically confers some sort of enhanced survival mechanism. Think of a plasmid as something that these bacterial cells can swap from one cell to the next. This gives me an advantage. Let me give it to you, and you use it to grow. A lot of times what grows with this is going to be antibiotic resistance genes. So hey, I've got the ability to grow, and you're going to die. So let me give you this plasmid. They have extra copies of their own. Let me give you one, and now you can grow and not die off, okay? So a plasmid is something that's going to typically help give an additional factor to these organisms, but it's something that kind of be traded like candy, like Pokemon cards, if you will, okay? They're really valued by the cell, but not every cell is going to be required to have it. Make sense? All right, so let's stop here. Let's take a 10-minute break, and let's reconvene in the lecture room.